get started. I am delighted to introduce to you Dr. Kerry Nickols, who is joining us from right up or down the road, still not quite sure. Still learning my California geography at Cal State Northridge, who's going to be talking about a topic that is, I think, near and dear to both of our hearts, which is kelp. So, I am going to go ahead and turn it over, and we are delighted to have you here and so excited to learn about kelp. Thank you so much.
You're welcome. Thanks for having me. So yeah, I'm a faculty member at Cal State Northridge, although this is my last week of full-time employment at CSUN, and I'll end with telling you about what I'm doing next. But the work in my lab, so I teach, but I also have a very research-focused position, and I've been doing a lot of work trying to understand how kelp forests along the coast of California could mitigate ocean acidification and hypoxia. And the work that I'll talk about today was funded by the National Science Foundation, University of Southern California Sea Grant, as well as the California Ocean Protection Council, which is a state agency that does a lot of oceans work. Get some pizza!
Yeah, oh yeah, come on in, get some pizza.
You only have 15 boxes of pizza to eat. So you gotta get started.
I think it's like one per pizza per person at this
one
point.
Pizza per person, for sure. So a lot of what I do is motivated by the rising concentrations of carbon dioxide in our atmosphere. So this is a figure from Scripps Institute of Oceanography, which runs the longest running record of atmospheric carbon dioxide measurements. And this is showing our current, the latest reading, which was taken just a couple days ago. It was 418.47 parts per million. And this record is going back, showing you before the Industrial Revolution, where carbon dioxide levels were and how that they've increased. And to put it in another perspective, if that isn't shocking enough for you, then here's looking back the last 800,000 years. So the really far back data comes from ice core data, and then the more recent data are the atmospheric measurements that Scripps make. So concurrent with rising concentrations of carbon dioxide, we have warming, which is probably what you've heard a lot about in terms of climate change, but maybe less commonly known by folks are the impacts on the oceans besides warming. So has anybody, raise your hand if you've heard of ocean acidification. Okay, so some people. So the carbon dioxide is going in the atmosphere and it equilibrates with the ocean. So we're also increasing the amount of carbon dioxide in the ocean. And that actually changes the chemistry of the ocean and it makes the oceans more acidic. Does anybody use a soda stream? Just like an old person. Young people don't use soda streams. I use a soda stream.
I do too.
So basically, like, it's a way for you to get sparkling water on demand instead of like buying a bunch of cans. So you take a cylinder of water and you inject carbon dioxide into it. So that's essentially like what we're doing under the ocean, except maybe a little bit slower than like the soda stream does it. So the oceans are incredible because they have absorbed 30% of the carbon dioxide that people have emitted. So like, thank you ocean, because we'd be like way worse off if like the ocean hadn't done that for us. So, but what's happening is it's actually making the oceans more acidic. This is a time series from this, from Mauna Loa, which is on top of, it's in Hawaii. And this is the same kind of data that you've seen before, but it's, I think these are like yearly averages. So this is the carbon dioxide data in the atmosphere using this axis here. But then what happens in conjunction, this line here goes with this axis, the PCO2. So this is the amount of carbon dioxide that's in the ocean. So it's not like the same amount, but you can see that it's also increasing. And that data comes from Station Aloha, which is an area where they do long-term measurements of carbon dioxide in the ocean. And then here's the record for pH. So, and I'm not going to show you the chemical equation, we're not going to go into it that deep, but basically like carbon dioxide makes the waters more acidic. It increases the amount of hydrogen ions in the ocean. So we see pH goes down. So more acidic means lower pH. And this is a problem because there's lots of organisms that calcify, that make shells, like oysters, mussels, some species that are really ecologically and economically important here in California and throughout the states. And they won't be able to make shells anymore. And once it gets low enough in terms of pH, their shells will actually dissolve. So people are pretty concerned about this, especially people in the aquaculture industry. They're like on it. They know that this is a massive problem that we're going to face and that we've already started to face. So these are model predictions from the latest report from the Intergovernmental Panel on Climate Change that came out just in 2021. So when we increase carbon dioxide, we're decreasing pH. That's also known as ocean acidification. So this is the actual data of how we've seen the decrease in global ocean pH. And then these are different model scenarios, depending on if we're like business as usual carbon dioxide emissions, or if we do something about it. But there's a lot of variability in terms of what you get out of these different scenarios. And this might not sound like a lot, like going from 8.05 to 7.7, but pH is on a logarithmic scale. You know, like the earthquake that just happened, right? 7.8 or 7.9, like, wow, really big, logarithmic scale. The difference between like even a 7.6 and a 7.8 is like really intense. So same kind of deal here. So in addition to this issue with the oceans becoming more acidic, we also have a problem where the oceans are containing less oxygen. There's lots of different reasons for this, but one reason that's relevant to climate change is that warmer waters hold less dissolved gases. So they just like physically can't hold as much oxygen in them as the water's warm. And we're seeing an increase in expansion of areas around the world that have low oxygen. So this figure is showing you these like different oxygen concentrations, whether they're below 2 milligrams per liter, which is when they start being considered low oxygen, which is hypoxic, but there's also areas that have are even lower than that. There are a lot of different plans and ideas from managers here in the US, like trying to think about what are we going to do about these problems with ocean acidification and hypoxia. And actually, we're pretty lucky to be in California, where California is doing a lot on the climate change front, and particularly in the oceans. And I was involved in this working group where we were trying to understand if organisms that photosynthesize could be used as a management tool to lessen the severity of ocean acidification and hypoxia. And the reason that people are thinking about this, and this is also mentioned in the US plan, but the reason people are really interested in this is because there are these organisms, you know, like the trees outside, that are absorbing carbon dioxide and releasing oxygen. Well, we also have those organisms. They're not actual trees, but we have plants and plant-like organisms in the ocean that are undergoing photosynthesis that could decrease carbon dioxide in the water, thereby increasing the pH and also increasing oxygen. So the organisms that might do this are things like seagrasses, which we do have some in California, and then also macroalgae, and in particular kelp. So this is giant kelp, which is the main species we have in Southern California. Get some pizza?
Yeah, eat pizza. One per person. Like one pizza per person. Yes,
one
exactly.
pizza per person.
Grab a box.
Exactly. Exactly. So I've been really focused on thinking about this question of if the submerged aquatic vegetation can lessen the severity of OA, ocean acidification, as well as hypoxia. And let's back up a little bit and get into kelp forests. So again, this is Macrocystis pyrifera, giant kelp that's found off the coast of Southern California. So has anybody seen a kelp forest before? Yeah. So if you don't think you have, you might have. If you ever go to the beach and you're like standing on the cliff and you look down, especially in like the Palos Verdes area or in Malibu, up in those areas, and you just see like brown on the surface, you're probably looking at a kelp forest. And they are these awesome systems that provide a three-dimensional habitat throughout the water column. So they grow from the rocks on the bottom up to the surface. And they are what we call a foundation species because they provide habitat for other organisms. And they really change the habitat around them as well. So they modify the habitat. there are a lot of ecologically and economically important species that live in kelp forests, and they harbor large amounts of biodiversity. They are also one of the most productive ecosystems on Earth, like per area. So they rival that of terrestrial forests. So because they are so productive and they have a lot of biomass, they are a species that people have looked into to think like, well, maybe they could do something to change the water chemistry. And here, just to give you an idea, because this last photo is really showing you what it looks like when you're in the water, when you're with these fun blue rockfish here. But when you're on the surface, so it doesn't just stop growing once it gets to the surface, it continues growing out and it creates these massive canopies. So again, like lots of material that is photosynthesizing, taking up carbon dioxide, and releasing oxygen into the water. Kelp forests are associated, especially this particular species, are associated with areas that upwelling occurs at, which is this really awesome phenomenon that happens along the California coast, which is why our ocean is so productive. So along the coast of California and in a lot of these other areas that are highlighted, especially along the coast of Peru and Chile and South Africa, and off the coast of New Zealand, we have winds that blow parallel to our coast. It's not quite as noticeable in Southern California as it is in Central and Northern California, but we get these winds that blow parallel to coast that are pushing on the surface water, and then because of the Coriolis effect, they get deflected to the right, and it actually pushes water offshore. But you can't just have this like hill of water offshore. You have to have water replace the surface stuff that's being pushed offshore. So water comes up from the deep, and we get these really cold, nutrient-rich waters along our coast. And here is what that looks like. This is a sea surface temperature satellite image from along the coast of California during a time when there's upwelling happening. And so these bluish colors are the really cold water, so 10 degrees Celsius, which is like 40 something, it's cold. And so, you know, in Southern California, we get like a little bit of upwelling going on here, but not quite as much as we do along the coast of Central and Northern California. But one of the interesting things about these upwelling regions is that when you get that cold water, that water is coming from deeper depths, and it's naturally also lower in pH and lower in oxygen. So it's kind of this interesting problem of like, we have these overall changes happening to the global ocean, but then we also have an ecosystem that already regularly sees intrusions of cold, low pH, low oxygen waters. So some of the organisms have evolved to live in these conditions, most likely. And then, you know, one other issue that's going on with kelp forests is because we've had a lot of marine heat waves globally, so we've had a lot of periods with anomalously high waters, we've seen decreases in kelp forests over time. And in particular, sort of side note, but just want to point out over here, Tasmania is this island off the southern coast of Australia, and I was just there for a conference that focused on temperate reefs, so all like basically cold water reef areas. So it's a lot of kelp forests. People go to this meeting, and I'd say about 80% of the talks that I saw were about heat waves and how they've decimated kelp forests around the world. And in particular in Tasmania, they've lost 95% of their kelp forests due to warming. But not just warming. A lot of these areas are getting sort of like, they're getting it on multiple fronts. And so in addition to warming, that sometimes can allow herbivores to come in and get a hold, and then they like mow down the kelp forest. And urchins are one of the primary culprits of that phenomenon. And they literally create these areas where you go from a kelp forest to an urchin barren, which is just urchins and like nothing else. So this is an image. These are a couple images that were taken where I was working off the coast of Monterey. So this also has been happening in California in some areas. So it's also important to understand what kind of services kelp forests provide for us before they all like turn into urchin barrens, which probably won't happen right away, but is something that people are pretty concerned about. So again, returning to like my big sort of research question is trying to understand if submerged aquatic vegetation like kelp can mitigate ocean acidification and hypoxia. To really look at this question requires being interdisciplinary and to think mechanistically about what's going on. If you just go out to the area and take a water sample and measure the chemistry, you will learn about the chemistry in that particular area, but you won't really know a lot about like the importance of kelp or what's driving the patterns that you're seeing. So my lab group and myself have really been looking at the biology and the physics and the chemistry of these habitats to try to understand how it all works together to really answer this question of what's going on. And so my background is in both earth science and ecology, and so, biology and ecology. So I've kind of always been this like creative thinker who likes thinking about holistic systems. And so first I'm going to share with you what we've learned about some of the physics associated with the kelp forest, and then we'll talk about the chemistry stuff. So the ocean is a three-dimensional fluid habitat. So, well, actually air is also fluid. We're living in a three-dimensional fluid habitat, but water has different properties, and it's more dense and it's more viscous. And when you have something that is throughout the whole water column, it's going to affect what the water's doing. And we know that when kelp are in water, that they change the way that water moves and the way that water is mixing. And so there's a lot of different processes going on in the ocean in terms of how it moves. And then the kelp can actually decrease the amount of currents. It decreases turbulence in the water. And this is also happening as like the bottom also has an effect on decreasing currents. And then that's looking at sort of like on the plant scale. I put plants in air quotes because they're not actually plants. And then if we look at a whole kelp forest, all of those plants together also work to change the way that currents are moving. So my colleagues and I did some work that was trying to look at how kelp forests impact currents. And one of the ways that was important in how we were thinking about this is that a lot of people have taken sites where they go to a kelp forest and they sample inside the kelp forest and outside the kelp forest and compare it. Or they go to one site that has kelp and another site that doesn't have kelp, and then they make measurements. But one of the issues with that is that there are probably other differences between those sites, like the rocks and the way that the coastline is, that can change the way that the water moves. And so we had a couple different scenarios off the coast of Monterey, where we've done a bunch of work, and then locally here off the coast of the Palos Verdes Peninsula, where we had an opportunity to measure the same site with kelp and without kelp and see how the currents are affected. And the reason that this is important when we think about the chemistry is because if you can imagine that the kelp needs some time to change the water chemistry. And so if the water is just like sloshing in and out, then the kelp isn't really going to have time to do anything. You'll just get new water coming in without the kelp modifying the chemistry. But the longer the water sits in an area, the more time for kelp to actually change the water chemistry. So off the coast here in Palos Verdes, there's this site that this big kelp restoration project was going on. So there was this pretty severe urchin outbreak and this massive urchin barren along part of the Palos Verdes Peninsula. And so the Bay Foundation, which is a local nonprofit, they got some funding to actually do manual urchin removals and to try to restore this kelp forest back to a healthy kelp forest. And they were successful in doing so, and it also created a really unique opportunity to look at how the water is moving in this area and how that changes as the kelp comes back. And so here are some data just showing you this. These are, we measured the density of kelp. So each one of these little strands is called a stipe, and we measured the density of the stipes. Starting in 2016 and going on past past 2017. This work was led by a woman named Kristen Elsmore, who was a PhD student at the time at UC Davis, who now manages the kelp plans for California, for the California Department of Fish and Wildlife. So we went out there and we measured kelp, and we saw clearly when they were doing the urchin removals that there was no kelp in this area. And then after the urchin removals were finished, the kelp started coming back. So there's this sort of transition time when we were moving from no kelp to some kelp. And then the kelp forest stabilized at that point. So now we're going to zoom in on that transition and kelp area, and I'm going to show you some data about how the water was moving during this time. And so we've got time here on the x-axis, and then we're looking at velocity on the y-axis. These velocity measurements are taken using these really cool instruments called acoustic Doppler current profilers that send sonar throughout the water column. And there's enough particles in the water that you get this, like, the particles bounce off of the sonar. And from the backscatter, you can actually measure how the water's moving, and you can do it throughout the water column. These data here are averaged throughout the water column, so they're averaged by depth, so that we just get like one measure of the current speed. And so we're looking inside the kelp forest is in gray, and then we had a similar measurement at a deeper area outside of the kelp forest in black. But in this part here, there was no kelp, just bare rocks. And then you can see as the kelp starts coming in, that these velocities get a lot lower, and they are significantly lower than the currents that are measured outside of the kelp forest. So we can compare inside versus outside the kelp forest to get an idea, especially without the kelp, of just how the change in depth impacts currents. Then once we get the kelp showing up, we can think about, well, how much additional reduction in velocity do we get? So one of the ways that we can quantify that, so taking that time series and then looking at it to try to see, like, well, what degree of reduction do you actually get? And what we're plotting here on the x-axis are all of the offshore velocities. And then on the y-axis are all of those velocities inside the area where the reef is, where you would have kelp growing. And these are all taken, all of these data come from the time when the kelp wasn't there yet. And so if they were the same, if you had the same amount of velocity inside versus outside that rocky reef area, they would all fall on this red line, this one-to-one line. But they don't. The velocities are decreased over here. So we get a 49% reduction in velocity that's just due to bottom friction. It's just due to going from like a deeper water column to a shallower water column. And then if we compare that to what's going on when there's kelp present, and again, here's that one-to-one line, you can see that there's very, very low velocities inside that kelp forest compared to outside of the kelp forest. So there's an additional 45% reduction in velocity attenuation that's due to the kelp. So this was a really cool thing looking at this restoration site. I also had kind of a sad opportunity to look at how the physical environment goes the opposite way. So when you have a kelp forest and then you go to an urchin barren. And this was at a place where I was working off the coast of Monterey that had this really interesting setup. So this is a pretty fun drone image of Hopkins Marine Station, which is Stanford University's marine lab. It's right next to the Monterey Bay Aquarium, if anybody's been to Monterey. And on this point here, there is this very clear area that is wave protected, and then this other side is wave exposed. And I'd been working there for a couple of years. And this figure is showing you the differences in wave energy, which might be a little bit hard to see, but the blue ones are from the wave exposed, and they're generally higher than the waves on the wave protected side. And so the wave protected side, I was made some measurements in 2013 to 2014, and then came back to do more work there in 2018. And when I came back in 2018, so before there had been a kelp forest that went kind of all the way around, and then when I went back, this was still a kelp forest, but the wave exposed site didn't have kelp anymore. And you can actually kind of see it from this drone image, like all of this dark stuff here is kelp, and there's like really no like dark stuff over there. And this image was taken in 2018. And I had current meters out, the same kinds that we use down in LA, we also had up in Monterey. And so these are the same kinds of plots that you saw last time, comparing offshore to the shallower area. And this is from the wave protected site. And this is with kelp, so in 2013. And then this is the wave exposed site in 2013, which also had kelp. So it's interesting that these two different sites, they actually had similar amounts of kelp at that time in 2013, but they had pretty different hydrodynamic environments, and the water moved differently there. So in the more wave exposed site, you actually didn't see as much change in velocity from the kelp that you saw at the more wave protected site. And then looking at 2018, when there wasn't any kelp, you see it actually falls pretty much on that one-to-one line. So you didn't really see any reduction in velocity in that area, which was pretty interesting. So you could imagine then, if you are in a kelp forest and you're trying to think about what's going to happen with the chemistry, how the water moves is going to be really, really important to figuring that out. And so my group and my collaborators have done a lot of work to try to show that the hydrodynamic environment is really important to consider. And a lot of the studies looking at chemistry in kelp forests haven't really considered how the water is moving, but that's like kind of a big problem. So now we can think, so now I've tried to show you that the kelp really are a really big part of the story when we think about how the water is moving. And then that's going to be really important when we start thinking about the physics. But just how important it is kind of a big question that we've been trying to look at. Because what ideally we would want is that somebody could look at a map, and if they knew something about the shoreline orientation, how wave exposed it is, that they could make some kind of prediction about is that going to be a site that might mitigate the chemistry or not. So I'm going to show you chemical data that come from two different sites. So again, the same ones I've already been talking about. So up in Monterey and then down off the coast here in LA. And here's that image showing you those sea surface temperatures to show you that these are pretty different sites also. Here's the Monterey site. So it's in this area that experiences a really high amount of upwelling. It experiences those cold waters, which sometimes are low in pH and low in oxygen. Down here is the Palos Verdes Peninsula, and the story is a little bit different. So the water's not that cold, and that will be important later on. In Monterey, my team and I, we've done kind of an insane amount of data collection, and most of those data will probably never be fully analyzed. That's the way it goes. But we tried really hard to measure both wave protected and wave exposed sites because we knew that the hydrodynamic environment was super important. So in 2018, we had gone back and measured that site along Hopkins Marine Station that I showed you before, and then in 2019, we sampled this other wave protected site and this other wave exposed site. And without not going to go into the details because I don't have time to, but we measured lots and lots of things, and so we had a pretty insane effort measuring chemistry. At one point, we had this boat moored in a kelp forest for 72 hours, and we would take 8-hour shifts, just being on the boat, continuously pumping water and measuring all of these chemistry signals, really trying to understand the carbon cycle in the kelp forest. We also deployed lots of instruments along what we call mooring lines. So this is a weight stack, and then there's a line going up, and then we put instruments on it, and it has a buoy at the top so that the line stays taut. So that's most of the data that I'll talk about today come from those types of oceanographic instruments. We also measured a ton of stuff about the kelp, how dense the kelp was. We measured kelp growth, where this originally had a tiny, just a little hole punch made there, and then we'd go back every week and we would measure how far the hole moved to measure the growth of the kelp. So we measured individual blades. We also measured the length of these stipes. Those are some of the coldest, hardest dives I ever did in my life because it was just really cold. And you would kneel on the bottom and you're like, 1 meter, 2 meter. And then you're like, oh wait, I lost count. And then you have to go back and start over. But we measured lots and lots of different things to really try to get a handle on kelp's capacity to change the water chemistry. So here are some data looking at pH. And actually, I'm going to back up a little bit. So if you see a biological signal from the kelp, like what might that look like? When we think about what's going on with photosynthesis, it's gonna, like the plants or plant-like things like kelp need light to photosynthesize. So photosynthesis is only happening when there's light outside. So you would expect to see a very clear daily signal in some kind of chemistry, like in either oxygen or maybe in pH, if there is a really strong importance of kelp. Because that's what the kelp is going to be doing. It's going to be doing photosynthesis, so you'd expect to see a daily signal. So these data here are daily averaged data that have also been filtered to be smooth. So we're looking at kind of a longer term signal than a daily signal. And you've got time here on the x-axis. This one is pH, and you're maybe looking at this and going, what is going on? Like, what is happening with these like decreases in pH, and then it goes up. We've got pH on the bottom, and then we've also got pH closer to the surface. Well, this figure here are the winds, and these are the winds that are driving upwelling. So you tend to see where you get strong winds, and then it plunges, that this kind of like matches up with what's going on. When you get prolonged winds, you get this dip in pH that persists, and then it kind of starts creeping up. So it really does seem like the overall signal from the pH is really driven by like what's going on at a bigger scale. And it's these upwelling winds and what's going on with upwelling. And not really shown in this figure, so these are data that came from inside of a kelp forest, but we also found during this deployment that there wasn't a lot of difference between inside and outside of the kelp forest. So that also made us think like, oh, that's really what's going on with the winds. But this is, you know, again, looking at it from like a day to sort of 3-day time period that it's being smoothed over. So if we look at what's going on at a higher frequency, so if we're like, what's going on in a given day in the kelp forest, that's what we're going to show here. This is, so you're kind of looking at a snapshot through the water column. There's time on the x-axis, and then this is the depth on the y-axis. This panel here is temperature. So you can see that what happens is there's cold water at the bottom and warm water at the surface. Then we get a lot of mixing, and then we get more cold water coming in. That is what's happening here. Also, look, this is velocity, and it's velocity that's going from outside of the kelp forest inside the kelp forest. And so you get these crazy things called internal bores, which are underwater waves that like come into the kelp forest, and they're driven by the tides. So this doesn't really have anything to do with the kelp forest. It's just like, here's this wave of cold, low pH. So this is the same kind of setup as these other plots, but looking at pH. And the blue color is cool, it's low pH. So you got low pH waters associated with these like cold water waves coming in. So at the first pass, it kind of looked like kelp's not really doing a whole lot. And really it's just driven by like the physics and what's going on in the ocean. But when we first made these measurements, we weren't really looking right in the canopy. We did a lot of work looking throughout the water column, but especially my collaborators were afraid to put sensors right in the canopy because they can get tangled in the kelp. And in this area in Monterey, there are also a lot of otters, sea otters, that like to kick it on the surface, and everyone was afraid that they're gonna like rip the sensors out. But as one of my collaborators, Yui Takashita, who's at the Monterey Bay Aquarium Research Institute, he said, well, anytime we put something in the water, we should just expect that we're not going to get it back. And he was like, let's put stuff in the surface. So we made these fun little floats to like keep the sensors in the surface, and we had pH sensors and dissolved oxygen sensors. This is one of my former graduate students, Demi Panos. And so, yeah, we have these really cool data that no one had really collected before, which seemed kind of shocking because you'd think you'd want to put the sensors where most of the kelp is if you're really trying to look at the kelp effect. So this is probably the most confusing thing I'm going to show you, but I'm going to walk you through it, so it's going to be okay. For each of these plots, we're going to eventually have more of them. We've got hour of the day on the x-axis, so it's just looking at like one day, so between midnight to 11 p.m., 11:59 p.m. And each one of these thin lines is a day's worth of data. And then the solid lines are the average of all of the days of data. These sensors were up for like 6 weeks. And on the y-axis here, we're looking at dissolved oxygen saturation. So basically 100% means that the water is fully saturated. If it's below that, then it's like undersaturated, meaning that there's like not as much oxygen in the water. And then it could be super saturated because the kelp are producing lots of oxygen, and well, just things in the water in general. We're looking at the surface in the bluish colors, and then below the surface, so like a couple meters below the surface. And so this is outside of the kelp forest. So at this particular site, and this was at a wave-exposed location, there's like maybe a tiny blip of a daily signal that would be due from like the microscopic plants that are in the water, the phytoplankton. But not much going on, and really not a lot of difference between the surface and the subsurface. In that same area, though, inside of the kelp forest, we see this really clear signal from the kelp where photosynthesis is going up, dissolved oxygen is increasing, and so we get this peak in dissolved oxygen concentrations that then, as it starts getting darker around 6 p.m. and so, then you see like that dropping off. And there's a clear difference between what's going on at the surface and what's going on a little bit below the surface. And we saw a similar thing for pH. So this was all dissolved oxygen, but we see the same story with pH. And we didn't have a subsurface sensor here. So this is on the wave exposed side, and we did a very similar thing on the wave protected side. And so here's our outside the kelp, which looks pretty similar to what was going on outside the kelp at the other site. And then here's our wave protected inside the kelp, and we see an even greater amount of, especially in dissolved oxygen, in this wave protected area. So we saw the highest pH in dissolved oxygen at the surface, so comparing the surface to the subsurface, and then a strong daily signal that's indicating the importance of the kelp canopy. Plus, we also saw interesting biogeochemical differences, so differences in especially the oxygen concentrations, and some in the pH too, between the wave exposed and the wave protected. So here's looking at the longer time series. So that was just looking at it one day at a time, but all the days on top of each other. Here's looking at a couple months of data for, and these are daily averages of dissolved oxygen. So we've got inside the kelp is solid lines, outside the kelp are dashed lines. Wave exposed is blue, and wave protected is red. And again, we see like higher dissolved oxygen in the wave protected side most of the time compared to the outside. So we tried to look to see what's driving these differences. They were similar depths and similar amounts of kelp in general. So we also looked at phytoplankton concentration. So we looked at like how much chlorophyll's in the water. That didn't matter. We actually looked at the community composition of all the phytoplankton. So we would take these water samples and then look at them under the microscope and try to identify the different phytoplankton species that were there. And this is like a crazy plot. But this is looking at basically how similar the different communities were, and they're organized by different sample dates. So all of the red ones were collected on June 12th and June 14th across locations inside and outside the kelp forest, as well as wave protected and wave exposed. And the thing to just notice here is that we didn't find it didn't matter which site we were at, they all had very similar communities, and the ways that the communities changed were just by date. So over time, we saw different communities of phytoplankton, but when we compared like the wave exposed and the wave protected sites, they had the same communities. We also looked at things like nutrients, we looked at kelp density, and we looked at kelp growth, and nothing explained the differences that we were seeing between our different kelp forests. But our kelp forests differed in their wave exposure. And so we had the higher waves and then the lower waves at the wave protected site. And so we're pretty sure that that's what was driving the differences that we saw. And so we did see higher pH and dissolved oxygen in kelp forests, and we saw higher pH and dissolved oxygen at the site with lower wave energy. However, the differences that we saw were actually pretty small. So there was only an 8% increase in dissolved oxygen outside of the kelp, and the mean pH was 0.05 pH units, which, you know, it's like, it is a difference. But the question then is like, is that at all meaningful for any organisms? Like, is that a big enough difference for like an organism to really care? And the differences between the sites were also pretty small. So the mean dissolved oxygen was about 6% higher at the wave protected site compared to the wave exposed. So kind of on par with the inside-outside differences. So then the next thing we started looking at was like, what's going on? There's also understory algae. So there's a lot of algae that live like on the bottom, kind of amongst the kelp forest matrix there. And so we thought like, well, maybe there's also potential for those communities to make a difference. And so the graduate student that I showed you the picture of earlier, Dimitra Panos, she did a lot of really cool work trying to look at oxygen like right in communities and then 1 meter above communities for different algal species and in an urchin-barren as well. And I'm not going to show you all of her data. I'm just going to highlight a couple things that we saw. So in Monterey, she also did this work both in Monterey as well as in Palos Verdes. And so in the Monterey area, there were 2 different understory communities, but their differences in oxygen were like minimal, and they really followed that signal of upwelling that I was showing you earlier. And for comparison, so this is data that came from the bottom, and then this is the data that came from the surface. And it's interesting that you see these like pretty clear daily peaks that come from the canopy that you just don't see at the bottom, even though you're in this like community that's also photosynthesizing. So in this area that has high upwelling in Monterey, the kelp signal is really confined to the surface. It does not go throughout the water column. And this is kind of a bummer because it means that the location of greatest amelioration potential, so like where you're getting like the greatest signal from the kelp in terms of the chemistry, is really disconnected from where the conditions are stressful. And actually, if I go back here, so this is that same kind of thing, percent dissolved oxygen saturation. This is saturated. So a lot of the time in this community, the water column, like at the bottom, it has very low oxygen. So that might be kind of a stressful place for organisms to be living. So what about down in Palos Verdes Peninsula, down in Southern California? So again, we were down here in an area that doesn't experience as much upwelling and just generally has warmer waters. So here, Demi had 3 different communities, a brown understory, a coralline understory, and then also just bare rock. And there's, again, no difference between any of these communities. But this, you know, it is more of a daily signal than like what we saw up in Monterey. It's not really an upwelling thing. So what happens if we look at the canopy there? Well, it's actually like right on top of this data. So you can see it's like a little bit higher sometimes, but in general, they like match up really well. And to contrast our 2 sites, so here's up in Monterey, and here is down in LA. And having dove quite a bit at the site, I can also attest that it's quite wavy. Just like not a lot of waves, just like enough to be obnoxious. And so it looks like there is a kelp signal, but it's being mixed throughout the water column, which is kind of cool. So thinking about like, if this water was mixed throughout the water column, then maybe it would line up on top of each other. But it's also, there's a lot more nutrients, and it's a lot just more of a productive environment up in Northern California. And so you get like really high highs, but you also get the low lows, which is interesting that we don't really quite see down here in LA. So we've done some work trying to like think about kind of some take-home messages about different types of sites. We made this schematic that I'm not going to like go into detail with too much, but this appears in a document that went to the state of California to try to, say like, okay, if you're in a site that's like different high upwelling versus low upwelling, low wave exposure versus high wave exposure, like what do you expect to see? So in these types of areas, it's more well mixed, and so this is kind of like the site that we had in Palos Verdes. We haven't really looked at a site that isn't super well mixed to see what that looks like down here without strong upwelling. So anyways, this is just the kind of way that we're trying to take all of the different pieces of information that we have and piece it all together to make some kind of management recommendation. But kind of regardless, it just doesn't seem like it's going to be that important. So the greatest amelioration potential was at the surface, especially in that area in Monterey, but that it also really depended on the site-specific physical context. But it doesn't appear that like the kelp are really going to change the chemistry that much to save us. They're not going to save the organisms there. There's lots of other great reasons to protect kelp forests, but like this is probably not one of them. And so a better option is to probably stop emitting carbon, which we need to be doing kind of anyways. And so I mentioned at the beginning of my seminar that I'm actually leaving my position at CSUN. And so I've just kind of gotten to the point where I realized that the work that I'm doing and the research that I'm doing can be important, but is not actually like advancing climate change solutions. And we're definitely at a point where like that's what we need people to do. So you young people out there, like we're going to need you. We're going to need you to help us. So I'm joining an organization called Ocean Visions, which is a nonprofit that works exclusively on ocean-based climate solutions. And I start on Monday. And I'm going to be leading a new program on ocean climate, like thinking about how we can actually repair ecosystems while we're also doing some other things to dial back climate change. So Ocean Visions is a nonprofit, but it works with a large network of these other institutions. And our goal is to confront and solve big, complex threats to the ocean. And so we're trying to innovate and deploy these solutions to climate change. And so we are a network organization that mobilizes diverse disciplines, sectors, and geographies. And we're at the intersection between science, innovation, business, and policy. So it's also like industry people, it's not just scientists. And then we're looking for like where some of these priority action areas are, and then trying to really start like getting to a point where we can deploy solutions, because a lot of solutions are actually not scalable at this point. And so this is all motivated by the fact that what we're doing is not really enough. So here's looking at emissions over time, carbon dioxide emissions. And you can see that there was this like blip, this like small decrease during the main times of the lockdown. But then we went right back to having pretty high emissions all around the world. It looks like the European Union might be like the best at sort of like decreasing emissions, but there's like a long way to go. And so also fossil fuels are still accounting for 82% of primary energy use. And then we're also still seeing that like the world's 60 largest banks invested $4.6 trillion in fossil fuels since 2015. So there's still a lot going on in terms of that. So anyways, just quickly, basically like we're realizing that there are these tipping points. So there's this thought that we need to keep emissions to warming no more than 1.5 degrees, or definitely not 2 degrees Celsius, but we're already seeing tipping points in the ocean. And so in addition to cutting future emissions, we also need to be removing carbon dioxide, and actually all of the IPCC scenarios involve some degree of actual carbon dioxide removal. And the only way we're going to meet those targets is carbon dioxide removal. And so then we're also thinking like, is there some things that we could be doing to try to repair ecosystems to keep them functioning while we're getting a handle on these things that we don't have a handle on? And so I'm just going to skip ahead to my acknowledgements. So that's what I'm going to be doing, and I'm really excited about it. So if you're at all interested in this, you're welcome. That's my new email address if you're interested in that kind of stuff. And then lots of people were involved in the science work that I presented. And thank you so much for your time, and I hope you get enough pizza.
Keep eating. Thank you so much.